Okay, so this is where we left off the ribosome, the big machine that's actually going to do the business of protein synthesis. There are two subunits. Both of these subunits are composed of, in this case, the small subunit 1 ribosomal RNA, the large subunit 2 ribosomal RNAs. You don't have to know this, but the eukaryotic ribosome has 3 ribosomal RNAs in the large subunit, and then each of these subunits, in addition to the RNA component or components, is composed of multiple proteins. Why do we call them subunits then? Because this piece and this piece are pre-assembled when translation is going to begin. So yes, all of the proteins here and all the proteins here and the RNAs are made separately, but once they come together to start with, they don't fall apart until the ribosome subunits are too old and not used anymore. You have to know that the 23S, the number for that, and the 16S, the number for that, just because I'm going to tell you functions for them. And you need to know the large subunit's 50S, small subunit's 30S. These numbers are different for the eukaryotes, but you don't have to know those numbers. Yes. The large subunit here, this whole thing, this blob, is 50S. It contains a 23S ribosomal RNA, a 5S ribosomal RNA, and then a bunch of proteins. You need to know the 23S because we're going to talk about that's what's going to have the enzymatic activity that forms peptide bonds, and you need to know that the whole piece, the top of the hamburger bun, is 50S. Just so if you know, like the 50S is the big subunit and the 30S is the small subunit. And the S are just sedimentation velocity readings, so it's just arbitrary readings. This was before all the proteins that made up the ribosome were known. So now we can talk about the molecular weight of each of the individual proteins in there, but at the time that was not known. All right. So when the ribosome comes together with an RNA together, the large and small subunits form these 3 sites that I introduced last time. The A site, the P site, and the E site. It's A, P, E going right to left if the ribosome is moving right in a right-hand direction, and that's the way you can always think about it. The A site, the P site, and the E site are not just in the small subunit. They are formed together, so there's a A site, like a pocket, that is composed of both subunits here. The P is both subunits there, and the E is both subunits there. The reason why we want to think of it in this direction is because that shows how, and we'll see in the next few slides, how proteins are made. So new transfer RNAs bearing amino acids are going to come into the A site. The transfer RNA that has the entire polypeptide or protein being made is in the P site, and then once the transfer RNA loses its connection to an amino acid, it ends up in the E site and goes away to be recharged. So it's easier to understand coming up in just a little bit when I talk about how it actually works. But before that, I have to talk about reading frame. So it was determined in a lot of cool experiments that are described in your book that you'll want to read about over Christmas break, I'm sure, once the class is over. But it's actually really cool how it was figured out. How would you figure out that the words in messenger RNA have 3 letters apiece, if you don't have the molecular tools like the ability to sequence DNA and things like that we did. So Sidney Brenner and Francis Crick did this with a combination of a whole bunch of mutations that were made in a bacteriophage. Anyway, so here's this. If you start in reading frame 1, like if you start with the T there and make 3-letter words, it comes out just right. But if you don't start with the T, if you start in the reading frame number 2, the second one, it doesn't mean anything. If you start in the third reading frame, so not with the TH but the E, it also doesn't mean anything. This is a strong example because this is our words with our alphabet. Because almost every 3-letter combination codes for something, well, every 3-letter combination does code for something, just not all of them code for amino acids, then you can actually get incorrect things. So this is the 5' end of a gene. You don't have to memorize this. I'm just showing that. So here there's 3 reading frames, right? But we're only looking at one of the 2 strands. Now here is 2 strands, 5', 3' going left to right up there, 5', 3' going right to left down there. If you start in reading frame 1, so ATG is methionine, GGA is glycine, TTT is phenylalanine, TTC also phenylalanine, etc. This is the real reading frame for this protein, one of the proteins that I work on in my lab, from where it starts. But if the ribosome accidentally started reading here, TGG is tryptophan, you get all the way down here until you get a codon that is a stop codon. So very early in the protein, you would get a stop codon and the protein would be non-functional. Even if you didn't get that, you can see that all the amino acids are completely different, and so it will be a protein that's just junk, that doesn't work. If you do the third reading frame, it is full of stop codons all over the place. Amazingly, if you do reading frames 4, 5, and 6, so this one, like lysine is AAA, like reading right to left on the bottom there, because it would be 5' to 3'. If the bottom strand is the coding strand, here's your 3 other reading frames that you would get. Amazingly, none of these have any stop codons in, which is kind of surprising. However, also none of them code for the actual protein, and so it will be junk. So there's 6 possible reading frames, 3 on one strand and 3 on the other strand, and it's a big job for the ribosome to figure out where to start. Now, really, the ribosome only has to pick from these 3 reading frames or these 3, because the ribosome is going to be working on RNA, so there would only be 1 strand that was transcribed. But in DNA, there's actually 6. Yeah, question? So when you're talking about DNA replication, it's somewhat, not want to say it's easy, but it's possible for the polymerase to make mistakes. Like it'll come to 3 T's and put in 3 A's, but it could accidentally put in a 4th A or miss one of the 3 A's or something. None of the genetics books that I've ever taught out of, or the molecular books that I've read, have ever talked about issues like this with ribosomes. So the ribosome doesn't have a problem, or else, I don't know. I mean, I've never seen it listed in any materials that I've ever read where the ribosome will like skip out of frame once it's in frame. The problem is getting it, the difficulty is getting the reading frame set right from the start. If it got set wrong, it would make gibberish, and there's no fail-safe. It would just make the gibberish protein, and eventually it would hit a stop codon and stop, and you would get this polypeptide that's just junk and doesn't really code for anything. This business down here on the bottom, where you can go through all those reading frames without hitting a stop codon at all, is pretty unusual because there's 3 different stop codons. So usually, if you're out of reading frame, you're hitting stop codons all over the place. Good, good? Okay, so this is where I'm telling you what you need to know, like for the exam. There's initiation, elongation, termination. We're going to talk about all those. There's initiation factors, a ton of them. There's elongation factors. You don't need to know the names of the individual initiation factors or what they do. That's for molecular genetics where we have a lot more time. Even there, I just go through the prokaryotic ones and not the eukaryotic ones. You don't need to know the elongation factors and what they do. You do need to know like the release factors when we get to the end, but there's multiple ones of those and we're just going to consider them as a unit. Well here, so you have to know what the initiation factors are, but not any of them by names. So the initiation factors are IF and then just numbers, and they help get everything set up. So in the prokaryote, and there's a difference between how the prokaryote and the eukaryote ribosome finds where to start. Prokaryotic ribosome gets set up with the small subunit, the RNA, and the initiator transfer RNA. So there's special initiator transfer RNAs that function to bring in the first amino acid that goes in every protein. There's only one codon for methionine, that's AUG. But for the first methionine in every protein only in prokaryotes, there's a special methionine derivative here called N-formyl methionine. It's just got a modification on it. It's sort of a signal that this is a methionine, but it's the one that's really supposed to be at the 5' end. Kind of like in eukaryotes, the messenger RNA will get that 7-methylguanosine cap. It's to show that the protein didn't get broken in half and a methionine just happens to be at the N-terminus, not the 5' end. So this is the initiation complex that gets set up. The small subunit interacts directly with the messenger RNA to set up the AUG start codon in the P site. And then the initiator transfer RNA comes in with the N-formyl methionine and gets that thing all set up there. So you can, this gets set up and it goes right to where the start codon is. And the way it does that is by using one of the ribosomal RNAs that I was talking to you about, the 16S ribosomal RNA. So we talked previously, I think, about, did we talk about ribosome binding sites? Maybe, maybe not. So ribosome binding site, RBS, microbiology people say Shine-Dalgarno sequence is right 5' of the initiator codon. This 5' to 3', 16S ribosomal RNA, part of it base pairs directly with the Shine-Dalgarno sequence, and that's how the small subunit lines up exactly with the P site over the start codon. So the way this gets set up exactly is that within this small subunit, in addition to the proteins that's there, is the 16S ribosomal RNA. So this interacts directly with that sequence. That's different than how eukaryotes do it, because in eukaryotes they don't have ribosome binding sites. So how's that work? In eukaryotes, there's interaction between 5' proteins that interact with the 7-methylguanosine cap and those poly-A binding proteins that we talked about. So this is not to scale, obviously. As we'll see later when we talk about translation in eukaryotes, we'll see that a cool feature of it is that the messenger RNA is actually circularized so that when the ribosome finishes, it can start again. The way this works in eukaryotes, and this drawing is not correct, cross out the large subunit. The way it works in eukaryotes is that the small subunit starts at the 5' end of the messenger RNA and then starts scanning down the messenger RNA. So not shown here is the initiator transfer RNA that has an anticodon that will recognize the AUG start codon. Right? So the small subunit is going to start here, and it just starts moving from 5' to 3' down the messenger RNA in the untranslated region until it finds the first start codon. And then it stops. And it stops there because the initiator transfer RNA will recognize the sequence of the AUG because it base pairs with it. And then the large subunit will come in and everything will come together. So do you see how there's a difference in the prokaryote and the eukaryote? Prokaryotic ribosome small subunit does not care where the 5' end is. It does not have to scan down the 5' untranslated region. There is a ribosome binding site in front of the AUG start codon. Indeed, in prokaryotic messenger RNAs, there could be AUGs up here somewhere, but it wouldn't matter because they wouldn't have a Shine-Dalgarno sequence or RBS, ribosome binding site, next to them. In eukaryotes, however, you would be in big trouble, so potentially big trouble, if there was a mutation up here that led to another AUG. Now there is a sequence that I don't have listed here but is talked about in the book that's close to the AUG called a Kozak sequence, and don't worry about it, I'm not going to ask you that. A Kozak sequence named after a scientist named Marilyn Kozak. But that's not as strong of a thing, I don't believe, as the ribosome binding site. So there's a chance if you have an AUG up here somewhere, the ribosome starts translating where it's not supposed to. Good with initiation? After both of these cases, once the small subunit lines up with the start codon, only then does the large subunit come in and the ribosome is whole. This is a protective mechanism because the peptidyl transferase center, or the enzymatic activity that catalyzes peptide bond formation, is in the large subunit. So until the small subunit gets set up at the start of transcription, the part of the enzyme that can actually join amino acids together is not there. All right, so once we've got initiation taken care of, then there's going to be a sequence of events that takes place over and over and over again once the ribosome starts moving on down the line and translating. This is the elongation phase. And you can see here there are these EFs, so elongation factors. They just help with the process. You don't have to know them for this class. So in each step, you will start with a transfer RNA bearing an a transfer RNA in the P site bearing the peptide chain. So in this case, its initiation has just started, but you can think of this as well as this could be in the middle of protein synthesis and there could be 200 amino acids hanging off of there. So pretend there's that in this case so you can see how it works. So then the transfer RNA, which is with whatever is the next amino acid, comes in to the A site. So remember, the P site is the peptidyl transfer RNA site. It's only got one amino acid here, but if it had a big chain, it would have the whole peptide on it. The transfer RNA with the next amino acid comes into the A site, which remember is the amino acylated tRNA site. So the site with the transfer RNA with an amino acid on it. Just like with DNA synthesis or RNA synthesis, the ribosome does not read these bases and know what amino acid to put into next. Which amino acid gets put in there is determined by the codon-anticodon base pairing. Only a transfer RNA with the correct sequence or with the wobble equivalent that we talked about can fit in here and get the two amino acids close enough. There's more to it than this, but this is what I want you to know for this class. Then, peptide bond formation is catalyzed by the 23S ribosomal RNA. And the way that happens is the elongating chain that is on the amino acid in the P site is transferred over to the amino acid in the A site. So that now the amino acid on the transfer RNA that is sticking in the A site here has the whole peptide on it, and the transfer RNA that's in the P site that previously had the peptide is empty. It has nothing. Once that peptide bond formation takes place, then the ribosome, small and large subunits together, is going to move 1 or 3 clicks down the line there. So one codon down the line. It's important to note that the ribosome is going to move from left to right on the screen here, but the transfer RNAs do not move relative to the messenger RNA. They stay base paired with it. So even though this is going to move from this transfer RNA is going to move from the P site to the E site, it will still be base paired with the AUG start codon. This process is called translocation. Not translation, translocation. So the ribosome moves one step down. Now this transfer RNA that formerly had the peptide on it was in the P site. It's now in the E site. E stands for exit. It's the easiest one to remember. And so when the ribosome moves down, this empty, uncharged transfer RNA is now in the E site, and it immediately loses affinity for the ribosome and goes off. And what happens is that it will get recharged. So this is for the start codon, so it'll get another N-formal methionine put on there, and then be able to come and start another one. If it was any amino acid in the protein within it, not right at the end, it would just get whatever amino acid that is put back on it. And now the transfer RNA that brought in the next amino acid, so it used to have just glycine, now it's got glycine at the 3' end and the whole rest of the peptide, or the C-terminus, and the whole rest of the peptide up here is now in the P site, the peptidyl tRNA site, and we're ready for the cycle to start again. So then whatever the next transfer RNA would come in here, the chain gets passed, translocation happens, the old transfer RNA that no longer has anything is removed, and the new transfer RNA that brought in an amino acid now will have the whole chain on it and be into the P site. Make sense? So it's just a process that goes over and over and over and over again. Because you said it made sense, oh yes, oh you're volunteering. So, oh yeah, sure, let's do it. So what's going to happen next? So here's your codon table. How to read so this is a table for the codons, not the anticodons. Got it? The codons are in the messenger RNA. So now we have the next codon here, ACG. So the first base is listed here, the second base is listed across the top, and the third base is listed there, but you don't even have to look at this because you can just look at the third base in the chart. So what is 5' to 3' ACG going to be? What's the first base is A, right? So now you're looking in this row. The second base is C, so now you're looking in this column. So this row, this column, so now you're looking here, and there's ACG. So it's threonine. And you don't have to know the names of the amino acids. You'll just be able to write THR right on the exam. Yes, question? Okay, so the way that we always write sequence for messenger RNA or transfer RNAs is going to be 5' to 3'. So if the messenger RNA here is 5' on the left and 3' on the right, what's the anticodon here is going to be what's over here and what's over there. Opposite. So 5' here and 3' there. So the 3 letters you said are correct, however, you were reading in the wrong direction. So the anticodon, the bases on the transfer RNA would be CGU instead of UGC. Right. You have to remember, the table is a codon table, not an anticodon table. So write that down. Tattoo it on the inside. Write it on the inside of your water bottle. Put a little sticky note on the bill of your hat. I'm not going to let you wear hats, so just so you know, I'm just kidding. Sure, why not? I mean, you can look it up, right? So now you've seen how it works. So the next one is ACG. So you're going to have a transfer RNA come in with a threonine amino acid on it. But I could also ask you 5' to 3', what is the anticodon on that transfer RNA? And then you would have to say CGU. And I will also put UGC on there because, got to keep it fun. So and I will mark it 5' to 3' as well. You just got to know that, anti-parallel, right? Good? All right, so we're starting to go, right? So a transfer RNA is coming in here. It's going to have an anticodon of CGU, and it's going to have a threonine amino acid on it. Oh, ACG. Threonine, yep. So a transfer RNA with the amino acid threonine comes in to the A site. Then what always happens next? What's the next step in the elongation process? Well, has that ever stopped anybody? And if people have been saying wrong answers the whole time, and it's okay, we figure it out. So before the ribosome moves, what has to happen? It's going to join the amino acids so that this chain that's on the amino acid in the P site is going to get moved over, so it's going to be N-formylmethionine, glycine, and then threonine over here on that transfer RNA, right? And this transfer RNA, that's in the P site is going to have what on it? It's going to have nothing on it, right? So once the transfer RNA with the next amino acid comes in, then peptide bond formation catalyzed by the 23S ribosomal RNA happens. That leaves the whole peptide chain now stuck to the transfer RNA in the A site, and the transfer RNA in the P site that formerly had the whole chain on it now has nothing, right? And that transfer, now the ribosome, it's all about, and we don't talk about it here, but it's all about a bunch of shape changes in the ribosome. So as soon as that chain gets moved over, then the ribosome's like, oh, okay, and it moves down 3 more. So then now the ribosome, the ACG codon will be in the P site, along with the chain that has the peptide on it. And this transfer RNA here, with nothing on it, will have moved from the P site to the E site, and it loses the affinity of the ribosome and floats away, and eventually it will get charged, or another glycine amino acid will get added to it. Good? It's just these 3 things happen. The transfer RNA with the new amino acid comes into the A site. Peptide bond formation happens. That moves the peptide or protein chain from the amino acid in the P site onto the new amino acid that's in the A site, and then translocation happens. It's 3 things. The new tRNA comes into the A site, peptide bond formation happens, translocation happens, and then it rinse and repeat, right? Still question? What's the question? We're good? All right. So that will happen over and over and over again, as long as whichever the next codon is actually a codon that codes for something. But remember, there are 3 codons here that are stop codons. They don't code for any amino acid. One of those 3 will be at the end of every protein after the last amino acid that's coded for by the protein. And then we go to the 3rd step. So we have initiation, where everything gets set up. We've talked about elongation, where you add one amino acid to the chain over and over and over again. And just like replication and transcription, you have the 3rd step, which is termination. So your ribosome, with all of the amino acids on it, reaches the stop codon, any of the 3, UAA, UGA, UAG. When this happens, the ribosome stalls. It stops there. The reason is pretty simple. Because there's no amino acid coded for by a stop codon, there's no transfer RNA that's going to be able to come in with that amino acid on it. So the ribosome just stops there. Then we're not going to need to know what the individual ones do, although the mechanism by which it works is pretty cool, I'm just going to say. The release factors come in, and they can fit into the A pocket like a transfer RNA would, and they do 2 things. The first thing is they cleave the polypeptide chain off of there. So this is already the last amino acid on the carboxyl terminus of the peptide. That gets cut off, and your mature peptide gets released. And then the ribosome has to move down one more step. So this actually acts like it's it sits in there like a transfer RNA, these release factors, and there's a mechanism to it. It's more complicated than what I'm saying here again. They cause translocation to happen one more time so that the ribosome moves down that way, 3 nucleotides. The last real transfer RNA is now in the E site. It comes out. Once the last transfer RNA pops loose, then the release factors come out and the large and small subunits of the ribosome dissociate from the messenger RNA. And then they are free to go back and start translating something else. One of the coolest things that happened in the 1990s, so I think it was like the late 80s or the early 90s, when self-catalyzing RNAs were discovered, self-splicing RNAs were discovered, and then scientists were like, well, what happens if these RNAs that are in the ribosome are more than just structural, and then by doing crystal structures, which is similar to the structure of DNA that was done, only instead of X-rays, now they use a beam of electrons, and the scattering of that beam of electrons will allow scientists to figure out the 3D structure. It was pretty cool to figure out exactly how the ribosome looks and works. And so I just put this in here to show you those three different colored molecules there are 3 transfer RNAs. So here's the one in the P site with the polypeptide on it. Here's the one in the A site that there's going to be an amino acid, and I don't see where it's shown there. And then here's the one in the E site that just hasn't been done yet. But it's a lot more nuanced than the hamburger bun diagram that is shown for ease of understanding. So the RNA sort of snakes through here. It's just like going to get pulled through there like that, and the peptide chain is just made like this. Then when you get to the stop codon, this is going to get cleaved off the last transfer RNA in the P site. Instead of another transfer RNA coming into the A site, those release factors come in there, chop off the chain, and then translocation happens one more time and everything falls apart. All right, so polysomes or polyribosomes. Remember we talked about during transcription that multiple RNA polymerases can be working on the same DNA to make multiple messenger RNAs at the same time. That is a way to amplify a signal. So you have one gene in prokaryotes or 2 genes in eukaryotes that are diploid, and you have to make tons and tons of protein. So one gene gets copied into a lot of different RNAs, and then each RNA is actually made into multiple proteins. And the ribosome does not have to go all the way down from the 5' to the 3' end of the messenger RNA and then fall off for another one to start. You get these polyribosomes or polysomes. So polyribosomes, shown here, exist in both prokaryotes and eukaryotes. But what is only in prokaryotes is what's shown here, right? In prokaryotes, you get what's called coupled transcription and translation. So because the messenger RNA is just messenger RNA in prokaryotes, as soon as that RNA starts coming out of the RNA polymerase, it can be translated immediately. In eukaryotes, you have to do 5' capping, do the spliceosome has to remove the introns, 3' poly A tail, and the mature messenger RNA then has to be exported out of the nucleus into the cytoplasm. Prokaryotes, there is no nucleus. There's no membrane-bound organelles. So as soon as, and also the RNA doesn't have to be processed, so as soon as it starts getting transcribed, it can get translated. So this is a cool example of that. So here's DNA. Here are the little ropes coming off, are RNAs being made. So a bunch of RNA polymerases are going from left to right there. making those RNAs, and each of the RNAs that's being made, you can see have those little dots on them. Those are not nucleosomes. They're not like the beads on a string. Those are actually ribosomes. So that shows how big the ribosomes are. This is double-stranded DNA, and these ribosomes are just those the huge blobs on there. So as soon as the RNA starts being transcribed from DNA, it can get translated. But in eukaryotes, you would have something like this as well. Only it would be that RNA would be circularized, like was shown in the picture previously. All right. Good. All right. All right. So let me move on. And again, to say that, pay attention. Like, look here. This is only 26 slides. If you look at the, for example, the slides for chapter 5, I think I had like 60 or more, like a ton more. So just know what I've got here, and you should be okay. So chapters 16 and 17, respectively, are gene regulation in prokaryotes and eukaryotes. So what we've talked about so far is when we talked about general mechanism of transcription. There we go. Just the general mechanism of transcription. Now we're going to talk about exactly how gene expression is regulated, including this really cool thing. So we'll see this again later on in the talk, but one of the great things about the last, like starting in the 80s a little bit, and like in the 90s and 2000s, but mostly in the 90s, crystal structures got a lot better. So we got to see exactly how all these little molecular machines work, at the level. I mean, this is a piece of DNA that's being bound here, and the reason I'm going to get, it's later in the slide, and the reason why these are able to bind is because, well, this little molecule here is actually going to make these two release their binding. But, binding of this little tiny molecule here changes the shape of this whole thing, this whole protein, to regulate whether or not it can bind DNA. And without the crystal structure, you would know that it happened, but you wouldn't know how it would happen because you can't see all these. So this is a ribbon diagram. So these are all those alpha helices we've talked about. And this is a good example. Here's one alpha helix, and then you go around here just non-structured, and then another helix over there, and then another turn, and then another helix, right? All right. And this isn't even the whole protein. So this slide is for, even though this is a prokaryotic chapter, this is an example of like there's a gene regulation occurs at many levels. And so in eukaryotes, the first example is whether the DNA is compact and not accessible to transcription factors, or whether it's relaxed. And we'll talk about in the next chapter how nucleosomes can move around and mediate that. Then also in eukaryotes, you get processing of pre-messenger RNA to messenger RNA, and then you're also going to get regulation, as I hinted at the last time, at the level of the protein. So you can get a protein that's made but gets phosphorylated to turn it off or turn it on, gets dephosphorylated to turn it back off, and also you can get destruction of the protein there. So we're going to step back from this complicated eukaryotic model and talk about the prokaryotic model in this chapter, and then go back to this in chapter 17. So in prokaryotes, the main contributor to gene expression is whether the polymerase can actively bind to the promoter sequence and start transcription. There's also some cool stuff having to do with the shape of the 5' end of messenger RNAs, but we don't have enough time to get to that in this class. I'm just telling you it's there, so this isn't the only level of regulation. Something additional that I haven't talked about before is that many prokaryotes use what are called operons. So all an operon is one transcription unit well, maybe they'll leave. One transcription unit that contains multiple genes. In this case, it's 3 different genes. And so because one promoter contains 3 different genes, you can turn on the proteins that make all 3 of these genes with just turning on one promoter or one switch. So why would you want to do this? In many cases, the proteins that are expressed there are for the same process. So we're going to talk about the lac operon. Sorry if you've had this in microbiology, you should know it, then you'll get it again. But there's, so that's for metabolism of lactose, a sugar. Also, for many of the biosynthetic pathways where there's like, for example, manufacturing a certain amino acid. And there are multiple, like the arginine pathway, I don't know, that we talked about. I don't know if this is the case, but it could be the case that those different enzymes that do the different steps in the pathway are all controlled by, by one operon. And in which case that would make things a lot more simple. You don't have to turn on 3 different genes. So you get one messenger RNA made that encodes 3 different products. So how is this accomplished? It's from what we talked about before. So in the messenger RNA that's made, you would separate the 3 genes by having at the beginning of each one 3 different ribosome binding sites. So there's a ribosome binding site here, one here, and one here. So ribosome would start here and keep going, and then somewhere over here there'd be a stop codon, and translation would stop. Ribosome would be bound here and start translating gene B, and somewhere over here there would be a stop codon, and translation would stop. Ribosome would bind here and start translating gene C, and at the end there would be a stop codon. This is different than eukaryotes. So with a couple of exceptions, which are actually, there's actually a special way it's done that's not like this. Operons are prokaryote only. So why would this not work in eukaryotes? Think back to what we just talked about earlier today. What's the difference between how the translation start site is found in prokaryotes versus eukaryotes? So here in prokaryotes, there's a ribosome binding site, right? That Shine-Dalgarno sequence or ribosome binding site is recognized by the 16S ribosomal RNA in the small subunit, and that positions the small subunit exactly where it needs to go. Does anybody remember how you're shaking your heads already. Does anybody remember how the eukaryotic small subunit finds where to start translation? Yeah, right, the AUG. How does it find it? So here in an operon, in prokaryotes, the 16S ribosomal RNA will interact with the ribosome binding site and line up. That lines the small subunit up so that the P site is right by the AUG. For eukaryotes, the small subunit also lines up so that the P site is right by the AUG in the messenger RNA, but it uses a different mechanism. What mechanism was that? What? Right. So the small subunit interacts with the cap proteins, gets on the messenger RNA right at the 5' end, and then starts moving from 5' to 3' and stops when it finds the first AUG. So that would be just fine for this one, the purple gene, gene A here, right? But gene B and C would never get translated because you would start at the AUG site here, but when you got to the end of the purple area, you would hit a stop codon, the ribosome would fall off, and how would it get back on over here? The only way the eukaryotic ribosome is able to find the start site is by starting at the very 5' end of a messenger RNA and scanning until you find an AUG. Right? And so that doesn't work for eukaryotes. There are a few operons in eukaryotes. C. elegans has at least a few, but in those cases, it's done differently. Once the messenger RNA is made that has multiple genes in it, special enzymes will do a splicing reaction where they will chop the genes apart, and on each of the genes that's not the first one, like this one here and this one there, B and C, they will put a fake 5' end on it. So then the ribosome can start at the 5' end and go in there. But that's different than this. This messenger RNA stays together all as one unit. And see, this is what I was talking about. In a biochemical pathway, you start with a precursor, and gene A is an enzyme that does this step. Gene B does step 2 from the square to the triangle, and gene 3 goes from the triangle to the, I would say diamond, but it's really just a square rotated a little bit. And that's how it works. So, in addition to the 5 subunits, 2 alphas, beta, beta prime, and omega that make the RNA polymerase core subunit, and also the sigma factor that helps that RNA polymerase find the -10 and -25 sequences, there are also regulator proteins. These are going to be generally either activators of transcription, which help turn it on, or repressors of transcription, which help turn it off. It's very important to understand that, so here's the gene here's the operon we're talking about. There's the promoter, there's gene A, B, and C that we just showed on the previous slide. The regulator proteins are going to be expressed in that organism's genome, but they are going to be expressed from completely different genes. Like here's a separate promoter that makes a regulator gene transcribed into RNA, translated into protein. This should not be understood to be close by. This gene for the regulator protein could be anywhere else in the genome, sometimes millions of bases away. So it just codes for it and makes it. All right, so there are 4 different mechanisms of operon regulation that you need to know. And I've broken it down into 2 main sets and then 2 subsets for each one. And if you listen to the definitions that I give you, then it will help you to figure out which one we're talking about. So operons start out as being either inducible or repressible. To induce somebody to do something, you have to convince them, right? If you induce something, so if a woman is past her due date and she gets induced for labor, they're giving drugs to start the contractions, right? So if you induce something, it wasn't happening, and now you turn it on, right? You can think of for inducible, you can think of inducing labor, and that'll help you. You know, you weren't in labor before, and now we induce it, and you are. So it's off unless the status quo is changed. Repressible is the opposite. It is what we call constitutively on. So it's always turned on unless it's changed to turn off. So it's on unless it's acted upon to get it to shut off. Then with each of these two, there is a positive and a negative. So there's going to be positive inducible, positive repressible, negative repressible. But instead of trying to memorize all those four, memorize what inducible, repressible are, and then memorize what positive and negative means. So positive means that an activator protein is involved, a protein that's going to turn something on. Negative means that a repressor protein is involved. So positive activator, negative repressor, that's easy to remember. And the other two are easy to remember too. So what the heck does all of this mean? Let's start out with a negative inducible operon. And there's a lot here, but I'll go through it. I'll go through it with you slowly. So because it is negative inducible, let's start out with inducible. What's that mean? It means it's off unless it gets turned on. And negative means that the regulation is a repressor protein. So it's off unless it gets turned on, and the protein that's involved is a repressor protein, and repressors turn things off. So a negative inducible always is binding to a repressor protein unless the status quo changes. So here are what are called the structural genes. That's just like the 3 genes in the operon. Here is the promoter. That's where RNA polymerase has to bind. Now I'm introducing another term called operator. That's just a sequence that's near the promoter, usually a little bit 3' to the -10 to -35 sequences. The operator is just a sequence of DNA that a repressor protein is able to bind. All right? So something we're going to have to understand to see how this works too is the concept of an allosteric protein. So if I talk about an allosteric change or an allosteric reaction, what that means is that the protein is capable of changing shape depending on if various other molecules bind to it. So you can see here, this protein has 2 different spots or binding sites in it, 2 different domains. The first domain right here is a DNA binding domain, so that is able to bind the operator sequence. So under normal conditions, the repressor is bound to the operator sequence and the gene is turned off because by occupying this position, RNA polymerase is prevented from binding to the promoter and transcription can't happen. Then you get the inducement. So remember, this is inducible. So there is an inducer molecule. Could be a protein, could be another molecule, whatever it is. It will bind to the second site here. So there's two binding sites. One's for DNA, one's for binding the inducer. The repressor protein is always bound to the operator sequence, but if the inducer comes in, it's better to show it up here. The inducer will come in here and bind to its site right there. When it does, an allosteric change takes place. The binding of the inducer molecule to its site changes the shape of the DNA binding site to make it so it can't bind DNA anymore. And the repressor will fall off as shown here. See, the inducer bound to its site, it changes the DNA binding site so it can't bind. So now the gene can be turned on. Nothing came in to turn it on. You're just relieving the repression. This is like when you're sitting at a traffic light or whatever, and light turns green. If you take your foot off the brake, car's going to go forward even if you don't step on the gas. Most cars, right? So there you go. All of these mechanisms are usually involved in a homeostasis. So you don't want this operon to be on unless the proteins it codes for are required. So usually the inducer molecule is going to be, as shown here, like some kind of a precursor for the biochemical pathway, right? So if this is present, or as we'll see with the lac operon, if the sugar lactose is present, then you want the genes. You want the genes here to make this product. We'll want the genes to metabolize lactose. So if the precursor is there, that will turn everything on so that it can get these genes expressed and use it up. Yes? Yes, it is like a double negative. So you can think of the inducer as repressing the repressor, right? So the inducer inhibits the repressor, which makes it so that it can't inhibit gene expression, right? The enemy of my enemy is my friend, right? Okay, so the other thing is it's a homeostasis mechanism though, right? So when that precursor protein runs out, you don't want to keep having the genes to make these products because there's nothing to act on. So as this starts to run out, it's a stoichiometric equation, right? It's chemistry all over the place, right? So this is not, this is not like this, right? On or off. It's like a dimmer switch. There's a whole bunch of those repressor proteins around, and there's a bunch of this. How much of the repressor is bound up and turned off? If there's a medium amount of the inducer, then maybe half is there and half isn't. If there's a lot of inducer, then all of the repressor protein will be bound up and the gene will be going full speed. All right. I don't want to go on to the rest of them. Look forward. So look through the stuff here. Read through all of these so that so that you're familiar with it before we start for next time. And I'll see you then, or I'll see you today if you have lab today.